Hello everyone. This is chapter 10, Introduction to Simple Experiments. In this chapter we're going to talk a lot about what makes this way of doing research using experiments helpful and in what ways there are challenges. So we'll talk about some examples of simple experiments. We'll define what an experimental variable is and the different types. Importantly, we'll talk about why they support causal claims. And then we'll go into some designs, both independent groups and within groups designs. And then at the end, we'll talk about the four validities like we've done with many of our research approaches. So first, the example is about taking notes. And this was done by Pam Mueller and Daniel Oppenheimer in 2014. They wanted to look at, does using a laptop help or hurt your retention, both of factual and conceptual concepts? So when they brought people in, they had them watch TED Talks on interesting topics. And when the people came in, there was either laptops in there for them to take notes on or notebooks and pens. When they came in with the notebooks and pens, that's what they would use, and laptop likewise. So the results are right here, showing both groups had a very small positive effect. So both laptop and longhand for factual knowledge. But when it came to conceptual, the laptop group scored really poorly, while the longhand group did very well. So in this case, everything about what the individuals experienced was controlled. They were either given a laptop or notebook and pens. That is one of the things that makes it an experiment. The researchers controlled their conditions. Okay, consider another example. In this example, researchers from Cornell in 2012 were looking at if an individual is serving themselves from a large bowl or a medium bowl, if they would, first of all, get more food, and if they would eat more food. And so there's just this communal bowl is either medium or large, and the participants would go into this lab. It kind of looked like a kitchen. They would serve themselves onto a plate. The researchers would weigh the plate before and after they ate, and they kept note of how much they served and how much they ate. And so the researchers controlled whether the participants got a medium bowl or a large bowl. The results showed that the medium bowl participants took less pasta on this left-hand side, and they ate less as well. So again, this is an example of an experiment because the researchers controlled the conditions that the individuals got. So when you're thinking about experiments, consider there's different types of variables inside of an experiment. So first is an independent variable. This is the one that you manipulate. So the conditions within the independent variable tell us the different groups. So for our pasta example, is either medium or large bowl were the conditions. The independent variable was the bowl size. The dependent variable is the measured outcome variable. So this is usually what we care about. Did what we gave them, the conditions, did it affect their behavior or their perception at all? And that's going to be the dependent variable. It's also good to keep in mind that there's control variables. And this is any variable that an experimenter holds constant. So for the pasta example, they kept the pasta was the same for both the medium and large bowl. This can be a really important concept. If we don't hold some things constant, then we won't be able to say what caused the change. So like I said, caused change. So experiments, this is what they're about. They look for causal claims. The reason why they can do this is they help establish covariance, so the relationship. They establish temporal precedence, and well-designed experiments establish internal validity as well. So what do I mean when I talk about established covariance? It's all about, is there a relationship between the variables? So is there a relationship between what group they're in and their score on whatever we're measuring? So what's helpful is we can get this relationship by answering the question, compared to what? And so to get covariance, we have to have some sort of a comparison to say, yes, this group did better than another. So often we're going to use different types of control groups. So the basic control group is just no treatment condition. They're not getting anything. The treatment group, there could be more than one treatment condition. So it could be different levels of the treatment, so a lot and not very much. And then there can be a placebo group where it looks like they're getting the treatment, but really they're not. So when there's differences across the groups, that helps establish covariance. Next thing it helps is that the cause variable precedes or comes before the effect variable. So the manipulation, the independent variable, comes before the one that we measure. So whatever that outcome is. Also, well-designed experiments establish internal validity. So when we're talking about this, we're going to have to consider design confounds, selection effects, and confound in general, which means confuse. So a design confound is when a second variable varies systematically along with the IV, or the independent variable, and provides an alternative explanation for the results. So you have a design confound anytime you can say, well, maybe it wasn't the medium and large bowl, maybe it was something else in there. Maybe the large bowl made pasta look really gross, and the medium bowl didn't, or vice versa. Systematic variability is what the problem is when you have a design confound. So systematic variability affects only one group, or it may affect both groups, but in different ways. So it may affect one group negatively, another group positively. Unsystematic variability is totally fine. It's just random and affects both groups in similar ways. And so it doesn't actually end up being an additional explanation of the results. For example, maybe the pasta experiment, instead of doing a lunch, they do a dinner. It's systematic if they only have one of the groups do a dinner and the other one does a lunch. If they both do dinner, then it's totally fine. The time affects the groups in similar ways. Another thing to consider that can get in the way is selection effects. So a selection effect is when a participant in a level of the independent variable are systematically different than the participants in another level. So anytime we let the individuals choose what groups they go into, so for example, if we have two groups and one is a candy group and the other group is a broccoli group, if we allow them to choose which group they're in, there's probably going to be differences. People that choose a broccoli group are probably going to be very different than one that chooses a candy group. To avoid this, we use random assignment. And that's where we just randomly assign an individual to one of the groups. It means they have an equal chance of being in any of the groups, and we control where they go. We also can use matched groups. So in small samples, sometimes the random assignment won't do everything that we need to. So sometimes a matched group can be helpful where when we assign someone to a group, we find someone just like them in terms of IQ, psychological flexibility, all these things, and put the other person in group 2. So basically person 1 and 2 are very similar, so the groups become more like each other. So when we talk about designs, there's many ways. There are tons of ways of designing experiments. The next few slides show a few of them. The general designs are independent groups design. Has a lot of names, obviously. And then there's also within groups designs. Independent groups designs is where individuals in one group are totally different than the individuals in another. Within groups design is where you often have, you're just looking at someone, the same person at one time point and another. So independent groups, it's different people comparing them. Within groups is comparing the same people, usually over time. So post-test-only design, this is where we randomly assign into one of the groups, so group 1 or group 2, and they have some sort of thing happen, some sort of experiment, and then we measure the dependent variable. An example would be randomly assigning to either laptop notes or longhand notes, having them watch TED Talks, and then taking a comprehension exam. So another one is the pre-test, post-test design. In this one, we randomly assign them into different groups. But in this one, we measure their score before, then give them the experiment, and then measure after. This one's great because you get to see the change from before they had the thing happen to after. Another one, repeated measures design. This is just where you have one group. They, for example, will taste chocolate with a Confederate, so just someone part of the study. And then they rate chocolate. And then they taste chocolate alone, and then rate the chocolate. If we do this, we might get results like this, where the shared experience, it looks like individuals like chocolate more when it's with someone else. These results were actually produced by some researchers, Boothby and her colleagues, in 2014. And so they use this, what's called the repeated measures design. It's similar to a post-test, pre-test, post-test design, except there's just one group and you're doing it over time. This one you could change this up too. We're going to talk about this, but you could have some people have chocolate with a Confederate first and then alone. And then other people have that switch where this is first and then this one, and see if maybe chocolate at time 2 is just not as great. Another design is a concurrent measures design. This one is where you just have one group and you're giving them two things at the same time. So in this case, a female face and a male face, where you have infants that are exposed to pictures of one and the other at the same time. And then in this case, they measured which one do they look at longest, which would be looking preference. So you're given two things, and either you explicitly make a choice, or with infants, you just record what they do. So the question may be, which design is better? It totally depends on the situation. So when it comes to your study that you're proposing, you need to think through what would get at the effect the best. So you may be interested in using a pre-test, post-test design. And if so, that's great, because it may answer your question really well, but there's things to consider as well, because you might have practice effects that by taking the pre-test, they learned something, they practiced it. Sometimes there's order effects, so if you're doing a repeated measures design, sometimes, for example, with a tasting chocolate with a friend or a Confederate and tasting chocolate alone, it might just be that tasting chocolate the second time just is not as good. And it has nothing to do with if they're with somebody or not. So we'd want to look at those things. Another thing to consider is carryover effects. So effects in which there's some form of contamination. So you have one condition, they experience it, and it doesn't actually leave. They hold on to it in some way, and it affects their scores in the next one. So there's ways to avoid the issues that come with things like pretest, post-test designs, and repeated measures and all these. One of the best ways of doing that is by counterbalancing. So you have the order be kind of randomly assigned to somebody. So one person might taste chocolate alone first, another person might do it with a Confederate first, and then it switches. We're going to talk about this a little bit more in a sec. So advantages of within groups designs. So first of all, individuals can be their own control because you're looking at where they used to be and then where they're at after. The designs also give you more power to notice differences between the conditions. And usually that means you require fewer participants than other designs. So the independent designs where you have independent groups, you're going to need more people in order to figure out the difference than within groups designs. So again, there's issues when you're doing anything that's over time. You can use counterbalancing like we mentioned. All of these things are things to consider if you're doing anything that has multiple measures across time. Another thing to keep in mind when you're doing this is that practice effects might just happen. So a lot of studies in schools, students are learning whether you're giving them this intervention or not. So sometimes just having within groups design isn't enough because you don't know if the students improve just because they're learning like they normally do, or if the intervention is actually helping. So I've seen this with reading interventions, where if they just have one group, maybe the students are just getting better because they're getting better. So I know we talked about counterbalancing a little bit, but I want to go into a little bit more detail about ways you can do it. So there's two types in general. The first is full counterbalancing, and this is where all possible orders are met. So when it came to the chocolate example, you could do taste chocolate alone first, and then taste chocolate with Confederate. And then the other possibility is taste chocolate with a Confederate, and then taste chocolate alone. There are no other possible orders that you could go in, and so we would have full counterbalancing. In more complicated designs, people will often use partial counterbalancing. One of those is called the Latin square, and that's basically that every condition gets to be at least in every possible order position. So every condition will be first at some point, will be second at some point, third at some point, and fourth at some point. Even if you can't do all possible combinations, it's good to use something like this so you can see each group at each time point. So like we mentioned, the disadvantages to within groups design, we talked about counterbalancing, but sometimes it just might not be practical or possible. Within group designs, you usually need to bring people back in, and if it's going to be very hard to contact the same people and bring them back in to experience the experiment, then it might not be a good approach for you. It's important to keep in mind that experiencing all levels of the independent variable changes the way participants act. Or in other words, demand characteristics, which occurs when participants pick up on cues that lead them to guess the experiment's hypotheses. And so at some point when individuals have been in lots of groups, they can quickly start to figure out oh, I see what they're changing. They might be interested in this. And that can actually change the way that participants act, which can be one of the design issues. So a question the book brings up is pretest, post-test, or repeated measures design. The book argues that they're not. Pretest, post-test has multiple groups, and it's just pre-experiment or experiment and then post. Where repeated measures is you expose them, measure, expose them, measure. In reality, you don't have to keep these so separate. You can combine them in some ways. You can mix and match just to meet your needs, whatever is necessary for you to answer your research question. All right, the last thing, interrogating causal claims with the 4 validities. So of course, again, we have construct, external, statistical, and internal validities. So construct validity is asking how well were the variables measured and manipulated. External is to whom or what can the causal claim generalize. Statistical is how well do the data support the causal claim. And fourth, with internal, are there alternative explanations for the outcome? So with the construct validity, when it comes to the dependent variables, consider how well were they measured. Do we think the dependent variables measured well? Second one, independent variables, how well were they manipulated? So often to do this, we need to do a manipulation check. And this can often happen in conjunction with a pilot study, a study that's really small just to see, does our manipulations work? So manipulation check, for example, if you were looking at the effect of fear on the ability to take an exam. So we might have 2 groups. In one group, we try to induce fear. Another one, we don't. And then we have them take an exam and report how stressed they were during it. So one thing we'd want to know is, did the thing that we were trying to do to induce fear, did it actually work? And that's what a manipulation check is. So we might actually want to measure how much did that scare you? Or we may look at heart rate if we don't want to ask them self-report questions. So we might look at heart rate and see, did their heart rate elevate during the fear manipulation. External validity, we always got to consider who is in our study. Does this represent a certain population? And if so, do we think that it will generalize to other people or to other situations? And the question is, what if external validity is poor? So remember that in an experiment, the validity that is emphasized most is internal validity. We're tightly controlling things, and so external validity sometimes can be really poor in an experiment. However, you can replicate the study in other situations and with other people to see if the results are very similar, then that supports external validity. Statistical validity is all about, did the data support it? And so you'd ask the question, is the difference statistically significant? And how large is the effect? So again, significance has a lot to do with, is there a lot of overlap between the 2 groups? Or is there less? So here there's a lot more, here there's less. So this one would be more significant, more likely than this one. This one also has a bigger effect size than this one. So this one would be more convincing of a causal claim in terms of it being an important causal claim than this one. This one, it may still be a causal claim and it may be valid, but it may not be as important. So finally, internal validity. So 3 fundamental questions to ask when it comes to internal validity. And this you can apply to your own design if this is what you're using. First thing, were there any design confounds? Are there any other explanations for the results? 2, if an independent group's design was used, did researchers control for selection effects using random assignment or matching? And 3, if a within group's design was used, did researchers control for order effects or practice effects by counterbalancing? So in your own study, be thinking about all these validities. These are things I'm going to be looking at when I grade your final proposals. I'm going to look at, well, did they address these different validities? Does it look like internal validity is strong, external, statistical? The statistical one is not going to be as important, and then construct is going to be important. So as you're thinking through it, keep all those things in mind. Go through each one and see, am I weak in any of these areas? And if so, look for ways to improve. So that is it for this lecture. Be on the lookout for Chapter 11 coming soon. Note that this week the abstract is due, but in just a few weeks the peer review is going to be due, which means you need to have your proposal put together so that it can be peer reviewed. I'll release another lecture about that, explaining it more, but keep that in mind.